All right, we'll go ahead and get started. Thank you everyone for coming to our Lunch and Learn. Today we have Dr. Kevin Coffey and his presentation on of mice and misuse, preclinical modeling of fentanyl use disorder. As you can see, this presentation is being recorded, so please stay on mute unless speaking and keep all private information about patients to yourself. And then for our next Lunch and Learn, we have Randall Espinoza, who is presenting on updates on neuromodulation techniques and applications. That will take place on February 26th from noon to 1 p.m. If you would like more information on that, feel free to contact me. In the meantime, we have Inga here who is going to introduce Kevin.
I'm Sierra. So I am pleased to introduce Dr. Kevin Coffey. He is a research assistant professor here at the University of Washington Department of Psychiatry and Behavioral Science, and is an investigator with the Mental Illness Research, Education, and Clinical Center at the VA Puget Sound. His lab aims to identify the neural mechanisms underlying substance use disorders and maladaptive decision-making. Dr. Coffey's current focus is on studying the neurobiological consequences and predictors of chronic fentanyl use. As many of you know, ADAI awards grants up to $30,000 a year to UW researchers who conduct pilot studies related to substance use. And in 2024, Dr. Coffey was awarded a small grant for a pilot study looking at once weekly GLP-1R agonist do I practice this?
Felicity, make it easy.
Dulaglutide for treatment of fentanyl use disorder in rodent models. Today, Dr. Coffey will talk about his work with preclinical modeling of fentanyl use disorders. Thank you, Dr. Coffey, for being here.
Yeah, thank you very much for having me. Yeah, I started my lab here just a couple years ago, but I've actually been with the department for almost a decade, and I have worked with ADAI both reviewing small grants and have been very fortunate to be a recipient of one of the small grants. You will get to see a small amount of data from that project today, and I'll explain where it's also headed. Yeah, so without further ado, let's just jump right into it. Make sure we can move. Great. Um, so I don't need to explain this to people in this room probably, but the United States is experiencing a pretty massive fentanyl epidemic. Fentanyl is an extremely potent and cheap synthetic opioid that is readily available. And in years past, it was really seen as an adulterant or something that was kind of being mixed in with other opioids of choice. But in recent years, fentanyl has really become the opioid of choice for individuals with severe opioid use disorder, particularly young people, people with comorbid psychiatric conditions. Um, and one of the really scary things about fentanyl is because of its extreme potency, it's very easy to overdose on. So I have been showing this graph, I think, in every talk I've given for the last 10 years, and I used to update it every year, and it just got, more and more depressing. So I stopped doing it before it took off the top of the screen. Fortunately, I think in recent years, or maybe even just the last year or so, kind of the dramatic upward trajectory of overdose deaths for fentanyl has begun to slow down. And I think that's, due in large part to a lot of hard work done here at UW through organizations like ADAI. And although the rise in overdose deaths has stalled, it still is a really massive problem and one that's very visible here in Seattle. And so what can we do to address this? Well, I think, I'll be the first to admit that pretty large societal change and healthcare and mental health work needs to be done. But I am a behavioral neuroscientist, so I work with rats and mice, and we still want to be able to contribute to improving the outcomes for people who are currently suffering from fentanyl use disorder. And so my lab really does kind of like three main things to try to tackle the opioid crisis in the ways that we can. So the first thing we do is we look at how does fentanyl change the brain. This is kind of the classic approach, this is your brain on drugs approach to addiction neuroscience. The things that we're trying to do differently now than they would in the past is we're trying to take a whole brain approach, trying to look at the brain holistically, what areas are changing, which areas are affected, and then we're also trying to take a individual differences and susceptibility approach to this because although many people throughout their lives will come in contact with opioids, whether it's through a surgery or even just trying recreationally, it's a pretty small subset of individuals that end up being extremely problematic users and at high risk for overdose, at high risk for interactions with emergency medical and criminal legal systems. So I'm going to talk about a pretty large collaborative R01 that does that looks at how does fentanyl change the brain. And then the other thing we're trying to get more into now than we have in the past is testing novel therapies, new therapeutics for opioid use disorder. And that's when we're going to talk about this new push to use GLP-1 receptor agonist, drugs like Ozempic, Wegovy, Trulicity, drugs that reduce food intake also seem to reduce intake of other substances, particularly alcohol and nicotine have been studied, and now we're going to try to look at that in opioids. And then kind of the other big thing that the lab is interested in is negative reinforcement in addiction. A lot of early addiction neuroscience focused on hijacking of the brain's kind of natural reward systems. How do people get hooked on drugs because they're rewarding, they're positive feelings the first few times. But a major problem with individuals who are in the throes of addiction is that negative reinforcement really drives continued use. So withdrawal avoidance, people really don't want to go through withdrawal, particularly for opioids. And then if they do make it through, stress is a really common driver of relapse. So we're really interested in these negative reinforcement mechanisms. So I'll talk about a few projects today, kind of give you a really high-level overview of what the lab does to tackle these three things. Okay, how does fentanyl affect the whole brain? So being here at UW is really fortunate. There are a lot of really amazing neurobiologists. I was fortunate enough to receive a pretty large R01. It's a collaborative R01 with four other labs actually, most of them now in neurobiology and neurophysics. Ling Kaigu does something called PixelSeq, which is a really amazing high resolution genetic mapping technique where you can visualize genes on tissue in large areas of the brain. Sam Golden does something called light sheet microscopy or whole brain tissue clearing. So he can take entire intact brains, make them see-through, and we can take pictures inside them and reconstruct them in 3D and look for where there are changes in brain activity. And then Nick Steinmetz is the developer of NeuroPixels, which are extremely high density electrophysiological probes that can record from thousands of neurons at the same time. So these are really cutting edge and amazing techniques to look at the brain. To apply this to fentanyl, though, we need to have a strong behavioral model, something that captures the different aspects of addiction that we're interested in. So that's really where my lab came in. And then John Newmeyer, who was my postdoc advisor, has also been helping out on this project. He's now my neighbor here at the VA, and we both are really interested in fentanyl and behavioral models of fentanyl. Okay, so the last year or so, I've been working a lot with two of Sam's, one of his techs and his postdocs, to develop a large scalable oral fentanyl self-administration model. So what does that mean? Fentanyl is a very potent drug, and so instead of having to give it to the animal through an injection, we can have the animals actually drink the liquid. And so you'll see on the right here is just a little video of one of the mice doing this. They're quite smart. They learn to press a lever, go get their drink of fentanyl, and go on. And they will do this for kind of like 3 hours straight, and over time consume more and more fentanyl. The main goal of this kind of early stage was to make this a scalable and accessible model. So what that means is we're trying to make it so that other labs, if they wanted to replicate this, could follow our instructions, and be able to build out kind of large behavioral models for relatively limited cost. And so the image on the left is just showing that we've basically turned these chambers into modular, scalable chambers that you can build, 3 at a time, and very rapidly increase the number of animals that you can run through this because, we're trying to get a lot of brains into the hands of the people that are doing the neurobiology. All right. So just to give you an idea about why we use this self-administration model. So what we're interested in doing is capturing the unique characteristics of fentanyl use that we see in humans. So one thing we see in humans is escalation. So these animals over time learn to take more and more drug. And then another thing we see is during abstinence, humans relapse. So we're trying to capture relapse in these animals too. And on the right, you see reinstatement. That basically means when we expose the animals to drug cues during abstinence, they start seeking again. So we can model both escalation, abstinence, and relapse in these animals. The other thing we wanted to do, like I said, is we're interested in individual differences, and a lot of the animal models use something called an inbred strain. So that means the animals are very genetically similar. So we thought we'd also test this with an outbred strain. That's animals that have a little more genetic diversity. They also have a little more behavioral diversity. So the data on the bottom is from a type of mouse called the CD1 mouse that's outbred. And you can see they definitely do not have the same kind of clean pattern that the C57s do. But that also gives us a wider range of behavior if we're interested in asking questions about, what brain areas are affected in the animals that are at highest risk or at lowest risk. Okay, and a little bit more data from this project. So when you run these sort of things, people are really curious, are the animals, like, do they know what they're doing? Do they know they're taking fentanyl? One thing that we do to ensure that that's the case is that we can change the dose on the animal without telling them. And they are very capable of understanding that the dose changed and then modulating their behavior in accordance. So these C57s, if you start to drop the dose on them every day, kind of by half-ish, what'll happen is that they'll have to press the lever more to get the same amount of drug that they did before. And so they're very, very capable of doing this. Just a good way to prove that these models are capturing kind of elements of addiction that we want, and that the mouse understands, what it's doing. It's pressing the lever because it wants to take a particular amount of fentanyl, not because it likes, lever pressing or drinking water. Okay, and like I said, once these models are up and running, we're really interested in individual differences. What's nice about these self-administration models is they give you a lot of details about different aspects of the behavior. So we can measure how much total drug the animal takes. We can measure how much seeking, like how much looking for the drug they're doing at the liquid port. We can measure how fast they associate the lever press to the drug, like kind of cue associations. And we can measure things about relapse and recall, all sorts of things that are relevant to addiction. And if you take all of the animals from a given study, and you take all that information, you can kind of condense the data into something called a PCA, or principal component analysis, where we project out each animal into this like addiction severity space. And this is really useful information, again, for down the line when we have a lot of neural data about what's going on inside these animals' brains. Now we'll be able to take that and map it back to this addiction severity space and say, in animals that are really taking a lot of drug or really susceptible to relapse, are there different brain areas that are active or inhibited, that sort of thing. Okay, so actually getting into some of that brain data. So this is a long project. The first year or so was, again, spent really honing in on the behavioral model. But now Sam's postdoc and tech have been working very hard to clear some of those brains and take a look at different brain areas that are active at different points in this process. And so what they actually do is they have a special machine that can suck all the lipids, the fats, out of the brain and leave the proteins behind. And then they can stain the proteins that are like they can visualize activity. So they use something called CFOS, which is an early marker of neural activity. And what we're doing here is just taking like a flyby through the entire brain, and every little dot that you see is a neuron that was active at the time that the brain came out. if you do this process with a whole set of brains, you can start to ask questions about differences. And so this data is really new, I think just in the last month or so. But to give you an idea, they can basically reconstruct the brains in 3D and look across, front to back, the whole thing, where are there changes? And so the data here is showing 90 minutes after a reinstatement or a relapse session versus 24 hours later. So when these animals are re-exposed to drug-associated cues, certain brain areas light up and other brain areas are inhibited. And again, this is really, really new data, so we're kind of just starting to poke around and see what we get. But there's brain areas that are, things that we would definitely expect, like the colostrum, areas like the lateral habenula, and areas like the raphe nuclei are active when the animal is processing these drug-associated cues. So just a little more pretty data because they gave it to me. Yeah, these techniques are really valuable for stepping back and not letting, the bias that, we all have about the brain areas that are most interesting to us. I think that's really common in addiction neuroscience saying, I like to study this brain area because I know it's going to be involved in this. Now we can take a step back and really look at the whole brain at once and try to open up new avenues, new opportunities for treatment down the line by pinpointing things that we didn't know about previously. Okay, so this part of the talk is going to be about our attempt to get into novel therapeutics. The lab has always been a very basic neuroscience lab. We study what goes on in the brain, but we have these models to have good, representation of fentanyl use behavior in animals, and so we can very quickly test new drugs to see if they're going to reduce intake. And one of the most promising classes are GLP-1 receptor agonists. So why are we doing this? So medication-assisted treatment, or MOUD, as it's often called, is really the most effective thing we have for treating opioid use right now. Uh, Buprenorphine, methadone, these drugs are very effective and much safer than taking street opioids, but they're not used as frequently as they should be. I think something like 50% of people who really should be taking these drugs have access to them, and I think in minority communities it's even less, something like 25% of people are getting access to them. And oftentimes it's because there's stigma, but also they can be inconvenient. Things like methadone, you have to show up in the morning before work, get it exactly on time in line, and then go out with your day. So there's a really big interest in expanding the pool of therapeutics to new drugs. And these GLP-1 receptor agonists came on the scene as very potent inhibitors of food intake. It turns out the brain also expresses a lot of this GLP-1 receptor, and some of these drugs make it through the blood-brain barrier. And so we chose to study dulaglutide, or Trulicity, because it is capable of penetrating the blood-brain barrier, and because we think it's acting on brain regions that are involved, in appetitive behavior. So in consuming things like alcohol and possibly in consuming opioids. So we have a very similar model to what you saw for the mice here, except we've extended it so that we can treat animals. A big thing we wanted to do with this project was treat animals who have already started taking fentanyl. So when these drugs were first being tested, they would give it to an animal and then ask, do they take more or less drugs? But we're not really interested in a preventative measure here. We're interested in something that could be, quickly scalable and given to people who are already in the throes of addiction, right? Like there's so many people, millions of people in this country that already need treatment. And so we need drugs that can be delivered at any time in their addiction cycle. And one thing that's really nice about these drugs is not only are they approved, but they're weekly and they can be given at home. You can take your shots home and maybe take them on Sunday, and hopefully it helps you the rest of the week. So that's how we designed this protocol. Animals take fentanyl for 2 weeks, and then we start giving dulaglutide on Sunday on the weekends, and it escalates over time. And so what do we see? Well, animals, just like in the last study, animals increase intake of fentanyl over the first 2 weeks. When you give that first dose of dulaglutide, you don't see very major changes. But when you give the second dose, suddenly we have our vehicle animals, the animals that don't get drug, continuing to escalate. This is really common. There's kind of a fast escalation early and then a light escalation after that. Our dulaglutide animals now have stopped escalating. This was the first study, the first run of animals that we did with this. It was very clear that they were kind of not escalating, but not clear that they were decreasing intake. And so a big goal in the future is going to be running these studies longer to see if basically this is the limit of the response in rats to make sure it doesn't affect weight. In this time frame, our animals do not lose weight, but it's also clear they're the same thing, not gaining weight, and rats usually gain weight quite fast. Um, and then we also have a lot of neural data from these animals, data from recordings using something called MiniScopes, and I'm going to talk about that a little more later. I would have liked to be able to start going over kind of what's going on in the brain, but it's a very technically complicated process, and we've actually spent the last year optimizing that on another project, and we now have kind of the staff and the expertise to take the data that's already attached to this project and start analyzing it. So hopefully if I talk to this group again, we can actually go through that. Okay. And then, yeah, the kind of the biggest part of our lab is looking at negative reinforcement in addiction. So what does that mean? I show this figure again in almost every talk that I give. This is a figure from one of George Koob's papers, and he has made like this figure over and over again in different forms. But basically, it shows kind of the affective progression, the emotional progression of opioid use. And this is from heroin, but it's very similar with fentanyl, and some would say even like condensed and more dramatic. So you take opioids the first time, they feel great, very like euphoria inducing, but that mental state quickly fades, and over time tends on a downward trajectory. And so there is this like shifting from the positive reinforcing effects of the drug now toward negative reinforcing effects, where the falling drug levels, the withdrawal all feel worse and worse, and so you have to take the drug to escape that. And then even if people do make it through to abstinence for a long period of time, they're generally very sensitive to stress, emotional distress, stress memories, and things come. This is the most commonly cited cause of relapse is stressful life events, where that negative mental state drives escape behavior through taking more drugs. And so we're really interested in that process. Um, like I said, a lot of neuroscientists have their favorite brain region. I am definitely in that camp. The lateral habenula is an ancient brain structure that's conserved in species all the way back to lamprey through mammals. It's highly conserved, and it's in our brains for a good reason. It's because it conveys really strong evolutionary advantage to be able to process negative events, to be able to process aversive events, to be able to escape from aversive events. It's a huge part of what survival is for animals. Um, Unfortunately, that means when things go wrong in the lateral habenula, it has really significant implications for anxiety disorders, depressive disorders, and addiction, because it is so potent of a brain region in processing those negative aversive events in life. Like I said, it's also there in the brain for a reason, and we need to make adaptive and value-based decisions based on negative information, negative reinforcement. And so it's not like something we can just knock out and everyone would be happier and healthier for it. It's something that we need, and we need to understand precisely what's going wrong in particular events like addiction to treat it. Okay, so one thing we do a lot is we model these different negative reinforcement aspects of addiction. One of them is withdrawal avoidance. So like I said, people really cite not wanting to go through withdrawal as a reason why they don't choose to become abstinent. It's a pretty miserable process for opioids. It's got really strong somatic symptoms. You feel sick, diarrhea, and then really strong emotional symptoms. I mean, like emotionally you feel terrible too. So in the animal models, what we can do as we can mimic this by getting them dependent on opioids and then giving them a drug called naloxone, or you may have heard it called Narcan. It's given to people who are overdosing. It very rapidly removes the fentanyl molecules from the receptors and puts people and animals into a pretty immediate state of withdrawal. And if we do this in a particular context, the animals will remember that context. They really have strong memories for, kind of where they were and what the world was like when they had this aversive withdrawal experience. And then we can test the animals and ask questions about whether they would like to avoid that environment that they experienced withdrawal in. And so we've done this quite a lot in the lab, but basically if you give animals naloxone after they were dependent on fentanyl, and then ask them whether they want to spend time in that chamber, they do not. They spend much more time in the saline paired chamber because there's no aversive memory associated with it. And so what we're interested in is what's going on in the brain when the animal makes these decisions to either avoid the chamber or leave the chamber altogether. We use a technique in the lab commonly called fiber photometry. What this does is let us put a glass fiber into the brain, a fiber optic cable basically, and we use a special virus to make the cells in the lateral habenula glow green when they're active. It's pretty simple actually. When the cells are active, they glow. That glow comes up the glass fiber, and we record it on a basically a fancy camera, a little photo sensor. And so what we're looking for here is what's going on when the animal is choosing to leave the naloxone paired side or enter the naloxone paired side of this chamber. And what we found was that the lateral habenula is actually active in both cases, which is not exactly what we expected. Basically, just the decision to switch chambers was being processed here. But the undergrad that was doing the video analysis at the time noticed that the animals also do something else. They make kind of rapid approaches to the transition point of the chamber and then stop. And this is really common in rats. They're very something called approach avoidant because they're prey animals. They're very nervous. They make quick movements to check something out and then they stop. And what we noticed is that when the animals are making quick movements toward the naloxone paired side, the withdrawal paired side, and then stop, we get a big increase in lateral habenula activity. But when they do the same thing toward the saline paired side, we don't see it. So it does look like there's some differential responding here, and that the lateral habenula is processing this decision to not enter the naloxone paired side of the chamber. We think that the data coming back from this is very variable, and that the habenula is actually a very heterogeneous region with a lot of different inputs and outputs. And so using this technique that measures kind of all of the cells at once is a bit fraught because you can add up things that are increasing and decreasing and miss important differences. And so we're working really hard right now to be able to record these cells at an individual cell level. And I'll probably get to talking to a bunch of that a little later in the talk. Okay. Like you've already seen, we do a lot of fentanyl self-administration. I wanted to bring this one back up again because we use rats primarily in our lab. We built the mouse model for the last project because of the tools that are available for doing neurophysiology in mice. That's what the other researchers needed. But I'm really interested in negative reinforcement and affect. And what the rat model allows us to do is listen in to kind of what the animals are saying. So rats are really interesting. They make something called ultrasonic vocalizations. So these are very high-pitched, above our hearing range vocalizations that sound a lot like bird calls when you slow them down. They're basically whistle-like calls. And they make a lot of these. They make ones that are associated with particular types of social interaction, different types of behavior. And they also make calls that are highly associated with their internal affective state. So when an animal is in a positive affective state, they make a certain type of call. And when they're feeling bad or when there's a reason for alarm, they make another type of call. And that's this 22 kilohertz vocalization. It's a very specialized type of call that rats make that's really useful for them to communicate with each other that either they're feeling bad or there's something dangerous around. And we can leverage that to ask questions about kind of how these animals feel. Usually in behavioral neuroscience, you're making interpretations from a lever press, or like I said, how much time they spend in one chamber or another. With rats, we can kind of drop that and let the animals speak for themselves, so to speak. So they will make these calls during fentanyl self-administration. And what's really interesting is that they make both types of calls, the positive and negative affective calls, but they make them in a very particular pattern. So like I said, when the animal first starts taking drug in the session, there's this really big rush. People that take opioids and then do self-report measures say the same thing. You get this big euphoric rush, but that is very short-lived. So in our sessions, maybe 15 minutes into the session, the animals will stop taking drug rapidly, and they'll start to transition to something we call maintenance, where they press the lever kind of, slowly and steadily to keep their brain drug level about constant. So at the beginning, big rush, they take a lot of drug, and then for hours they'll hold the that drug level constant. And during that time when they're holding the drug level constant, they really only produce the negative affective USVs. And so what we think is happening here is that every time the drug level drops, basically after every single hit, the drug level starts to fall. The animal experiences that feeling of falling drug level negatively and then takes another dose to escape it, to alleviate that. And that's very similar to what we would say with like negative affect emerging over time, but it actually happens within really individual drug binges. So negative affect is driving seeking in these animals kind of on a daily basis on every single time they're using drug. We, like I said, are interested in what's going on in the brain when these sort of things are happening. So we have used that same technique, fiber photometry, to look at the lateral habenula while animals are taking fentanyl. And what we notice is that when the animal presses the lever and hears a drug-associated cue, you often get an increase in activity in this region. And that increase is modulated by whether the animal is in this negative affective or positive affective phase of self-administration. So in the first 15 infusions, where it's all positive, euphoric rush, the habenula is not really activated so much by cues. But then for the rest of the session, when they're in the negative affective phase, we see really big increases in activity in the lateral habenula during the lever press. And so we think the habenula is, it's integrating information about the drug itself, about the cues, but then is modulated by kind of the aversive state that the animal is in. And then, really interestingly, when the animal consumes the drug, takes the drug, in either case, we actually see the habenula be inhibited, so it drops in activity. And so, I think is happening here basically is that these increases in habenula activity are driving some downstream motivational structures to, promote intake. When the drug is consumed, then the activity of the habenula falls off, and the animal feels better. They wait and take their next dose a little later. So that work was the basis for the R00 that is funding the lab primarily right now. What we're interested in looking at is if we inhibit those lateral habenula neurons during the times when they're active, can we reduce overall intake? Can we reduce the animals like fentanyl cue association? And can we reduce relapse to cues down the line? This is quite a large project, but it's being taken on by our postdoc Sierra, who is maybe in the audience right now. Sierra has a lot of work ahead of her, but basically what she's doing is she's injecting a special what's called an inhibitory opsin into the lateral habenula. This is something that you can use to block activity in the cells using light. So she's injecting that into the lateral habenula, and then she's putting an optical fiber into the brain regions that the lateral habenula projects to, and she's trying to inhibit them kind of one by one to determine if different populations of cells in the lateral habenula are responsible for these different aspects of self-administration. And so she has been optimizing this for the last few months, and hopefully we'll have a whole bunch of data in the coming months and I believe is taking the data to the SPEAR conference, which is a big neuroscience and emotion conference locally here. I think it's in Calgary, that quite a few of us are going to. So if you're there, I'm sure she'll talk about it. Okay, and then the final project I'm going to talk about is our work looking at fentanyl seeking despite negative consequences. So I think this is a really, common thing people understand about severe addiction is that, for a lot of people, like consequences start piling up, they change their behavior. For the people that's the most problematic use, consequences don't seem to be effective at reducing fentanyl intake. And so this is work in collaboration with Ari Asarch and John Newmeyer. Ari's one of John's grad students. What Ari has done has taken the self-administration model that we developed and modified it so that he can ask questions about how much drug animals are willing to take as escalation of consequences happens. So he trains these animals in a very similar way to us, but then in the later weeks, he starts introducing a shock, a consequence basically to every time the animal takes drug, and that consequence it accelerates over days. So it starts very small, and then over time it gets higher and higher and higher. And what we're really interested in is, the break point, the decision point, when an animal is going to continue to choose taking and when the animal stops taking. What are the differences in the brain that predict that? Can we harness those? Can we find those? All right, so what Ari has found is that as you increase the shock, there's not too much change in behavior early on. So a really small shock actually increases the total number of drugs into the drug that the animals take. I think these shocks are like very mild. If you've ever felt a 0.1 milliamp shock, it's kind of like the equivalent of licking the 9-volt battery. There's something about this stimulating effect of getting shocked. They're actually taking more. But as the consequences ramp up, eventually the animals will choose to completely stop taking. And so what we're interested in is kind of this, this inflection point, the decision point. What's going on in the brain? Oh, and I like showing this. This is just an interesting way to visualize what's happening. Every single tick on here is a response that an individual animal is making, and it's across time in the session. And so, like I said, at the beginning of the session, they're taking a lot of drug all at once, and then they begin to space it out through the rest of the session. You can see there's really not a huge change for these early shocks, other than the animals tend to kind of pack their responding tighter together during the shock sessions. I think that's because they're trying to trick the system. They might think that, if they can respond quickly all at once, maybe they won't, get as many shocks or something. They basically change the pattern, but they still take the same amount of drug overall. But by these late sessions with the high shock, the animal really is not responding a lot. And so we only have a few instances where the animal chose to take drug that we can then look at those instances and see if there's something different going on in the brain. Okay, and then just a really quick, Ari is also recording USVs in these animals. It's, you're seeing the same kind of pattern of positive affective calls early in the session and negative affective calls later, but on the low shock sessions, you actually, like I said, they were taking more drug. You actually see kind of a new peak in these positive affective calls, almost like the mild shock is interesting or novel to them. But by the time you get to the high shock session, there's this really huge increase in those alarm calls, those negative affective calls. So it's very clear that they're not happy being in that situation and are, finally so unhappy that they're stopping taking drug. Okay, so how do we ask questions about the brain in these animals? So I was showing fiber photometry data before. That's data from the whole brain region that we're interested in. But those brain regions are made up of individual cells, and those individual cells tend to process information differently depending on their genetics or their inputs and outputs. And we really, really want to be able to do single cell recordings of these animals. So over the last year or two, what we've done is tried to optimize a relatively new technique called miniature microscope recordings. So these are miniature microscopes. They are a complete fluorescence microscope that has been miniaturized to the size that it can actually sit on the animal's head. And then in our lab, what we've done is modified them to be able to withstand multiple hours of a rat on fentanyl with it on its head. You know, rats are actually very strong. You ever played with them? They'll run around, they smack into walls, they will break sensitive equipment. So we've had to optimize these things to be able to stay with the animal the entire time they're in these sessions. And then we've had to optimize the kind of the hardware that goes into the brain. So what you're seeing on the right are actually glass lenses. So these are basically like a microscope objective or a microscope lens, only they can be implanted deep into the brain. And then the microscope can look down that lens and actually see inside the brain at microscopic levels in real time. And so what happens when you do that? On the right here is basically an image of what you'd see through the lens. All the little dots here are individual cells. And then when you play this, what you'll see is like very subtle changes in brightness. And what we need to be able to do is isolate each one of those cells and ask, how is the brightness changing? And doing that is non-trivial. There are a lot of tools out there for doing it, but Ari, again, is a very dedicated graduate student and has decided that none of the tools suit his needs, and so has built an entirely new software platform called the MiniScope Processing Suite. This is new software that's graphic user interface based. It can take these images and even from really long sessions, gigabytes and gigabytes of data, and extract all the individual neurons, extract their signal over time, and let you ask questions about individual neurons changing during these self-administration behaviors. And then not to leave well enough alone, there's another issue with this is that we are interested in how the neurons are changing over days. And so that is also non-trivial. You have to be able to take these images and line them up and say, this is the same neuron on the first day of self-administration, on the last day. This is the same neuron on a low shock session and on a high shock session. And so Ari again decided, well, why don't we just build this all from scratch, and is just about to release a new software called Stars to Cells. What he very cleverly figured out is that there are very smart people working in astronomy that know how to map stars in the sky from different angles and different perspectives by using basically quadrilateral alignment. So they put a whole bunch of polygons up on the stars and then try to match those polygons across sessions. And he worked with some people that kind of knew how to do this already and built a brand new, again, beautiful GUI-based piece of software where we can put in each session and it will align all those neurons over days using this star tracking based alignment. And that way we can track the kind of the identity of a cell across our experiments and tell how is the processing changing. And so again, this data is really new. Ari just got this to me like this week for a grant. But basically what he's seeing is that over sessions, the LHB neurons are in fact processing things heterogeneously, so not all cells are doing the same thing, and that they dynamically reorganize across punishment sessions. And so this is a really small chunk of the data, but it highlights kind of how these cells are changing. So on the left is the activity of cells during some of the low shock sessions, and on the right is the activity of cells on one of the high shock sessions. And what happens is the activity tends to shift from the shock itself or from the lever press toward the danger cue. So as the animal learns more and more about what's going on in the session, instead of being surprised by the shock and we get a big burst of activity, that activity shifts backwards towards the cue that actually predicts the danger. And there are different populations doing different things. Some are still active during the lever press or like anticipation of the shock, and others are active again, like when the animal hears the predictive cue. And so what we need to do now is really dive in and look at what are the differences between trials where the animal chooses to take drug and trials where the animal chooses not to, and see if there's a different pattern of activity that we can then use later on for manipulation for treatment purposes. So what we, I mean, ultimately want to do is be able to promote the decision to stop taking drug without affecting kind of like the other things that the lateral habenula needs to do. So isolating individual populations is a really big strategy moving forward and the basis of kind of the R01s going out right now from the group. Yeah, I think that's enough. I have things after the slides, but I think it'd be a good time to say thank you to everybody. My lab is growing. We started here at UW in the VA only 2 years ago. It's kind of a tough time to be a new lab, particularly at a federal facility. But Sarah's worked really hard, and our new techs have been doing really great work. John, who has been my mentor for a long time, has been really instrumental in helping the lab get off the ground. And then all of our collaborators over at UW. It's really great to be in a community where there's opportunities for collaboration, things like ADAI, where we can get small pilot grants to go after these new things that will help the lab expand and find new focus in the future. So yeah, huge thanks to everyone who's involved in all this. Thanks ADAI for having me. And questions?
Uh, Thank you very much, Dr. Coffey. It's amazing to me how much you've done in 2 years. So does anyone have any questions right now? Well, is there more that you wanted to share? You said you had some.
Easy. I will just, yeah, I might as well. I'll just pop by and show you. So like I said, the cells in this brain region are doing different things, right? It's a really tiny area. This is something on the order of, like fractions of a millimeter cubed, but it's really important and it receives a lot of inputs from kind of frontal brain regions involved in emotion and cognition and like fear. All of these emotional things come into the lateral habenula and then they project out to regions that motivate behavior. And so what we're thinking is that the kind of segregation of activity is dependent on their inputs. And so what we're trying to do now is isolate those different cells. So one technique that we're using is something called intersectional or transsynaptic labeling. And so we inject a virus into one of the brain regions that projects to the lateral habenula, and then a second one that's dependent on receiving that input into the lateral habenula. And so this brain region, the lateral preoptic area, is really involved in processing like noxious stimuli, shocks, or like the really terrible feelings, aversive feelings of withdrawal. Those project to a certain subset of cells in the lateral habenula. And then other brain regions like the antipeduncular nucleus project to a different subset. And you can really see kind of they're in different spatial patterns, but they're also intermixed. And so we need ways to be able to kind of isolate those. One thing that we're doing is actually electrically stimulating those regions and then recording the change in activity in the lateral habenula so we can pinpoint exactly which cell is receiving input from each region. And by doing that, we can then try to manipulate those pathways in tandem with like understanding which, part of the fentanyl addiction behavior that they're associated with. So for us, it's basically withdrawal we're really interested in, and then relapse. And those two things seem to be governed by different processes within the brain. And so we want to be able to separate them and like target them differentially for treatment. The lateral habenula has definitely been seen as like this interesting new brain region to target for treatments of addiction, in anxiety, in depression. But if you target the whole thing, there's a lot of risk of causing issues with cognition, anhedonia, like adaptive decision making. You can't just take this whole brain region and shut it offline, right? Because it's going to cause a lot of side effects. And so the goal here is to be able to isolate very small subpopulations of cells that are doing one particular thing. And that way, if we do try to inhibit those, we don't screw up the rest of the processing. That's the long and short of kind of the future directions for this brain region in the lab. That's everything I put. Great.
There were just some comments, especially around the cross-pollination from other fields.
Yeah. Yeah, we're hoping to collaborate with other labs here at UW that have kind of different expertise. So there's labs that have been working on developing new drugs. Marco Previtoni is here at UW and works a lot with like vaccines, but is also interested in developing novel GLP-1s that are better at crossing the blood-brain barrier. So hopefully we'll get to a collaboration one of these days, maybe write a grant. Susan, got a question?
Yeah, really great talk. Fascinating information, especially about the emerging technologies that you're using to explore neuroscience of fentanyl addiction. I attended a really interesting talk a couple years ago at CPDD with humans who were reporting that their subjective experience related to fentanyl use, and particularly how it's kind of hit different, if you will. Now obviously, you can't ask rats about their subjective experience in taking the drug or their withdrawal from the drug. But I'm wondering if you could see any signals in the data that might suggest that fentanyl is hitting different, particularly in terms of maybe the negative reinforcement that's going on. Maybe it's particularly more aversive to withdraw from fentanyl. Like, can you speak to that at all?
Yeah, a little bit. So, like I said, we studied these vocalizations in rats a lot. They are a really nice measure of affective state, and we've done this years ago in cocaine and amphetamines, and then now through opioids. And I will say, I think the vocalization pattern in these fentanyl animals, to me, looks like more, I wouldn't say it's like different. There's definitely still the positive affective rush and the negative reinforcement side, but just they make more vocalizations. They're like also associated behaviors. So animals have these like stereotype behaviors when they're on drugs. They're pretty severe for fentanyl compared to something like morphine. They have really noticeable something called like a strop tail. Their tails actually stick in the air when they're high on fentanyl. It is definitely yeah. At the end of the day, it's an opioid receptor agonist. It's incredibly potent and yeah, incredibly fast acting. And those are the kind of things in humans that are like the reason why, methadone and buprenorphine work, even though they're opioids, right? Is that they're slower acting. They kind of like take those big peaks and valleys and they spread them out, and it makes people functional. It makes them able to, go about their day. Yeah, I can't see anything worse than just like straight up, straight down, straight up, straight down all the time. Really stressful on the animals and body.
Thank you. Really fascinating stuff.
Anyone else?
I have a question. So, and thank you again for presenting here. It is very interesting. So uh, with the people that we work with, sometimes or oftentimes there'll be a variety of drugs. So polydrug use, including combinations of opioids and stimulants. And I'm curious if on your roadmap, looking at those same brain centers with either alternating administrations of drug classes or simultaneous, if that is something that you're thinking about or have done, and uh, if that sort of changes or what that does to your thinking about this phenomenon.
Yeah, for sure. It's definitely on our, like on our radar. There are actually labs here at UW that specialize. I mean, Susan's lab, you're all familiar with Susan, has been doing that for a long time. She has animals on these heroin and amphetamine cocktails. Yeah, it definitely changes things. It's like it's another step in that, like scary direction of really fast ups and downs when now you're um, uh, adding stimulants on top of it. And then another really kind of terrifying one is people using xylazine as an extender. So, like you said, fentanyl is so fast acting, the come down is really fast. Other drugs like xylazine will extend that, but those have their own suite of like issues with like blood flow, and people have really bad issues when they're mixing fentanyl and xylazine. So it's definitely on the roadmap. Every time you add more complexity to the model, it can be harder to make, like the interpretations we want to make, right? So it's really interesting. There are people that here at the VA, Abby Schindler has like these naturalistic models where animals can choose different substances they want to take. So yeah, I think it's definitely happening. There are people doing it, and we'll keep it in the back pocket for when it makes sense for the right studies.
Well, if there are no more, if there, if there are no more questions, thank you very much, Dr. Coffey, for sharing your work. I look forward to hearing, after, and it's been more than 2 years, I look forward to hearing even more about your work.
Great. Yeah. Thank you for having me.
Thanks, everyone. See you next time.